So it's my pleasure to introduce to you today Dr. Karen Quigley, who's going to be our WKW Frontiers in Neuroscience lecturer for the day. Dr. Quigley received a bachelor's in zoology followed by a PhD in psychobiology from The Ohio State University in 1993. After completing a postdoctoral fellowship at Columbia University, she started her faculty career as an assistant professor at Penn State University in 1995, followed by faculty positions at Rutgers, the New Jersey VA, and the New Jersey Medical School. She moved to Northeastern University in 2010, where she is currently a professor of psychology and serves as the fellowship co-director for several training programs at the Bedford VA. Dr. Quigley has been funded by a variety of sources, including the NIH, both NIMH, the National Cancer Institute, the VA, the National Science Foundation, and the Army Research Institute. She is an elected fellow of the Academy of Behavioral Medicine Research and a fellow of the Association of Psychological Sciences. Over the course of her career, she's supervised numerous trainees who've gone on to establish academic careers of their own, including folks like Ian Kleckner, who previously conducted some collaborative work with LIBR investigators. Dr. Quigley is internationally recognized for her psychophysiological studies, having published more than 80 papers related to this area, for her service on the editorial boards of Psychophysiology and Biological Psychology, and culminating in her service as the president of the Society for Psychophysiologic Research in 2014. She's thus a card-carrying psychophysiologist, and I'm confident that you'll readily see the rigor and meticulous quality of her work in today's talk. So her work. Dr. Quigley's work focuses on how body regulation and interoceptive signals help the brain to construct emotion, including the role of individual and situation-level variation in emotional experience, and how emotional experience relates to behaviors, cognition, and health. She uses laboratory and ambulatory measures to understand what factors explain the enormous variation in physiologic changes, brain activity, or expressive behaviors that scientists observe across emotional states. Her recent approaches to discern emotion in the wild, as I like to call it, use innovative ambulatory and experience sampling approaches to derive real-world insights into the links between emotion and interoception. Now, I could go further into the details about her efforts to discern how interoceptive processing is related to integrating multiple sensory signals, or how changes in internal bodily states influence physical symptoms during major life stressors. But I'll pause here so she can take the stage. And please join me in welcoming Dr. Karen Quigley.
Thank you for that. Whoops, 1 second. Thank you for that lovely introduction. Oh, that was a little loud. I understand it's okay for me to be maskless during this, so it's easier to have a conversation, and I will be pretty well pinned here, so I won't be moving around spreading too many germs. It's really a great pleasure to be here. I have been trying to make this trip for some time, and I kept saying, no, I really want to come in person. I really want to see LIBR. I know from the work that's done here what an amazing group of people this is, and I just really wanted to see where you do your work. And so I kept pushing it off until COVID let me come. So I'm really glad to be here. I'm very honored. So today I'm going to say a little bit about why it matters that a brain is in a body, and of course I do feel a little bit like I'm preaching to the choir here. So my hope is that something here will be new to you, and I think the way in which we approach these questions might be slightly different than the way some of you approach questions, but it might be that we are also very kindred spirits. So I'm going to frame today's discussion from the perspective of predictive processing, or predictive processing view of how a brain and a body work together to optimize the functioning of a human. So you can think of the main job of a human brain is to make sense of the world, and it can only do this from inside the black box of the skull. And you think about the brain has to learn what's going on in the world only indirectly through the bits of information that come in through the sensory inputs. So sights and sounds and smells and so on. And the brain has to guess what's causing these sensations so that it can determine what to do about them. Now, another way to say this is that your brain is generating an internal model of your body in the world. And many neuroscientists now describe the brain's internal model in predictive processing terms, which I know many of you use this sort of predictive processing approach. Now, using this language, we would say that the brain is generating predictions about the likely causes of changes in incoming sense data. And when your predictions are correct, they become your experience and they guide your actions. And the brain keeps making its best guesses using past experience, and in this way, memory seeds future predictions. So how is it that the brain uses these noisy, ambiguous, incomplete sensations and makes some sense of them when all it really has access to are the effects? Well, we think that your brain is using prior experience to generate predictions about incoming sensory inputs well before they arrive to the brain. And keep in mind, sometimes we don't predict correctly, right? When this happens, a prediction error is generated, and that serves to correct the predictions. And in this way, a prediction error signal can alter future predictions. And from your brain's perspective, you can think of your body as just another source of information. It's just one more thing it has to make sense of. It's the sensations from your heart pumping, your lungs breathing or filling with air, your immune system, the metabolic signals from your gut, the fat in your body, etc. And these are all the interoceptive senses. This is the range of kinds of work that folks here are very intimately familiar with. So what you experience is then confirmed or modified by what's happening in the sensory periphery, both from the exteroceptive senses, vision, audition, etc., but also the interoceptive ones from your body. So here's my roadmap for today. First, I'd like to talk a little bit about how predictive processing applies to how a brain coordinates the actions of a body. And in this section, I'm going to define and describe predictive regulation, which goes by a term allostasis. And I'm going to talk a little bit about why a brain should operate predictively when it regulates its body. Then I'm going to describe how interoception or sensory signals coming in from the body are used by the brain to inform it about the state of the sensory periphery, along with incoming exteroceptive sense data. And finally, I'll share a little bit of data relevant to two implications of this view. One of them about how the body and the brain work together to coordinate affective experience, and the other one, very briefly, some preliminary data about how it coordinates for action. Now, Peter Sterling and colleagues compiled an extensive body of evidence showing that engaging in anticipatory or predictive regulation is how a brain maximizes its energy efficiency. Basically, it pays to prepare ahead for imminent action rather than wait to react, because it turns out that is slow and it's energetically inefficient. And evidence suggests that energy efficiency is the major concern for all organisms, right down to single-celled bacteria. And we know about anticipatory regulation, those of us in the room who are psychologists, because we've heard of the work of Ivan Pavlov, right? So remember Pavlov's dogs. They learned sensory cues specific to the context where food was going to appear. And after learning, they increased salivary flow, right, to prepare to ingest and digest the food that was going to appear. And in predictive processing terms, we would say that this increased salivation was driven by visceromotor predictions, right? These directed metabolic resources to where the dog thought it was going to need those resources next, namely in its gut. And human brains have been subject to the same natural selection pressures to optimize energy efficiency. In fact, a critical feature of allostasis is that the brain drives preparation for action via very specific and localized regulation, and it marshals resources to where they're going to be needed most. So what are some of these key resources that are required for allostasis? Well, biological resources have to be both renewed and removed. Right? Sometimes those are renewed by active foraging, sometimes by less demanding motor actions like breathing. So we breathe in oxygen, we ingest water, we ingest salts, we ingest nutrients like glucose and amino acids. And some energy is stored in the body for quicker access, and when the stored energy is broken down, we also are producing a lot of waste byproducts, things like carbon dioxide and urea. Those have to be excreted or exhaled back into the environment outside the body. And all of this renewing and removing itself requires energy. Now, measuring this resource exchange in freely behaving humans is a bit tricky. Because metabolic needs are specific and localized to different parts of the body. However, at a global level, we can try to at least estimate energy resource mobilization by measuring various features of the superhighway that moves those resources around. And that superhighway is the cardiovascular system. Now, allostasis requires fast, targeted control of blood flow, meaning that the cardiovascular system has to marshal resources to where the brain expects they're going to be needed to support action. And that means we have to try to understand multiple aspects of that energy delivery to the body. So the function of the pump, which is the heart, the pipes, which are the vessels, and the fluid itself, which is blood, right, which is carrying a lot of these metabolic resources. Now, the idea that the blood flow to the periphery is increasing and changing in supportive action is actually an old idea in my field of psychophysiology. Back in the 1960s, Paul Oberst called this cardiosomatic coupling. And we can see this during a motivated performance task. Now, by motivated performance, I just mean it's some task you've given a subject, they think it's self-relevant enough that they're trying, they're motivated, and they're willing to try to engage in creating an outcome and reaching some goal. And there are two general patterns of cardiovascular response to a prototypical motivated performance task, like giving a speech. This is always a fun one to show when you're actually in the midst of doing it. In one pattern, which we call a challenge response, we tend to see there an increase in blood flow, an increase in heart rate, but critically a decrease in total peripheral resistance. And what that means is that basically the vasculature is opening up slightly, if anything, and it allows really to mobilize a lot of fluid flow through the system. On the other hand, there's a very different pattern, which we see in other subjects or in other contexts, which we tend to call a threat response. Here you tend to see a decrease in blood flow relative to baseline, an increase in heart rate again, so notice that heart rate is not a distinguishing feature of these patterns, but also an increase in total peripheral resistance. So now the vasculature is actually clamping down, and what you do is you're circulating less blood flow than before, or rather that the increase is not as large as before. And this task really illustrates the idea that the brain coordinates these distinctive patterns of cardiovascular response that can support different bodily actions in the context of engaging in some sort of goal-based behavior. So if energy regulation is the core task of a human brain, then allostasis relies not just on moving resources to where they're needed, but also on feedback from the body, which reports on what's happening out in the periphery. And that brings us to interoception. Now, as this group well knows, interoception refers to these viscerosensory inputs that arise not just from the viscera, but also from endocrine organs, immune system, etc. And these viscerosensory signals inform the brain about the state of the body. And interoception you can think of as carrying some of the prediction error signals that inform the brain's internal model about something that wasn't quite correctly predicted. And more broadly, these ideas suggest that allostasis and interoception are at the core of mental activity, whether it's thinking, perceiving, behaving, remembering, even emoting. In a predictive processing sense, we can view all mental activity as arising from a brain that is constructing predictions in the service of allostasis and receiving interoceptive prediction errors as well as exteroceptive prediction errors from the world. From this perspective, it's useful to think of affect in a particular way as a low-dimensional feature that likely arises from interoceptive sense data. Now, two really common descriptors that you will have heard of about how to describe affective features are valence and arousal. Valence simply meaning how positive or negative is something, and arousal, whether you feel very, very activated or very deactivated. Now, interoceptive sense data is also noisy, it's ambiguous, and it's incomplete. And this does seem to be kind of a key difference between sort of quintessential or at least canonical interoceptive channels and exteroceptive ones. And it may be partly, if not largely, because viscerosensory inputs are carried along relatively thin, often unmyelinated fibers, and they're often also diffusely arranged in these organs. And it makes those signals slower, but also potentially more vague or ambiguous in their quality. And this means that any given affective change, to the extent those are carried by viscerosensory afferents, can have multiple possible causes. And so that means that the brain can have any one of multiple possible interpretations or predictions about what those vague sensations may mean. Now, in 2017, we reported a test of the hypothesis. This paper is actually, the first author is Ian Kleckner, who had come here to do some collaborative work. Ian and the rest of our group reported a test of the hypothesis that the brain systems or networks that represent the interoceptive or viscerosensory signals from the body and the allostatic or visceromotor control systems are linked, strongly linked. And to assess this hypothesis, we used seeds for all limbic cortical regions in the brain. And as we predicted, we found a system consisting of two overlapping resting state networks. So resting state networks, or what are called intrinsic networks, as you're probably well aware, are discovered when a participant is resting, doing no task-related activity. And these two networks that appeared actually have conventional names that we think of them more commonly as, which is the allostatic network we tend to call the default mode network, and it also goes by many other names, and the salience network, which seem to be more linked to the interoceptive network. Now, these two networks link in cortical hubs, and these cortical hubs are the most connected regions in the cortex. And these core hubs are also called rich club hubs, and they're thought to be the brain's backbone for communication and synchronization across all brain networks. And these networks contain portions of the insula, particularly the posterior insula, which, as you all well know, are the places where these interoceptive sensory data first enter. Now, it turns out these networks also have far more extensive subcortical projections than generally has been appreciated. And in fact, these networks are connected to the hypothalamus, to the periaqueductal gray, parabrachial nucleus, and even reach down to the brainstem, to the nucleus tractus solitarius as one of the main brainstem nuclei receiving a lot of viscerosensory afferents. So the suggestion here is this: whatever else these networks are doing, psychologically speaking, they're also regulating and receiving information from the brain's physiological systems. And this suggests that the body is a critical part of the brain's internal model. And it also suggests that we have to incorporate the body into our understanding of how human brains construct a predictive body, rather a predictive model of itself in the world. So if interoception matters for allostatically efficient organisms, like humans and every other organism, then we need to try to measure it. However, as this group well knows, it is tricky to measure interoception in humans. And there remains a fair amount of confusion about how to link interoceptive measures to interoceptive constructs. So I want to just very briefly describe two tasks that I'm guessing most of you are very well aware of, so I'll try to do that a bit quickly. But I'd like to sort of contrast these tasks because I think they tell us slightly different things, and it's the case that sometimes in the literature these two are used as interchangeable. And I'll try to illustrate why I think it's important for us not to use them interchangeably. Now, these are both cardiac interoceptive tasks, but there are versions that are somewhat similar that can be used in other systems, but I'm just going to compare today two, which are the heartbeat detection task, sometimes called a heartbeat discrimination task, and also the heartbeat tracking task, or sometimes called the counting task, the Chandri task. These basically are going to be sort of my canonical comparison points here. Okay, so first to the detection task. The version that I most commonly use, and there are many different versions of this, is that basically you're going to present a series of tones that are coincident with or non-coincident with a person's heartbeat, at least at the point at which they should feel it in the periphery. We do know there are individual differences in where people feel it in the periphery, and some of the techniques actually try to titrate in on that as best we can. And what we do is then we ask, after they've heard this series of 10, was that coincident with those tones, coincident with your heartbeat, or were they not? Okay? And the nice thing about this task is that you can then get a measure of sensitivity. So you can, using a signal detection theory approach, you can see whether someone is more sensitive or not. And you can also get a measure of bias. In other words, are they very likely to say something is coincident or not, sort of irrespective of what the stimulus is. Now, another nice thing about this one, so it has a couple of major advantages. One of them is that this is truly a measure of sensitivity, and it is not impacted by beliefs, and I'll say more about that in a second. Now, there are some major disadvantages of this task, however. First of all, it's extremely long, especially if you do enough trials to get a nice, reliable answer, and of course, subjects find it incredibly boring, right? So then you have subjects falling asleep, which is not so optimal. In addition, the task is hard, and it means that the number of people who are categorized when you at least do a kind of categorical separation, the people who are categorized as sensitive are relatively few proportion of the population. So that led to people using very commonly something called the tracking test or the counting test. So you have a start tone, they count how many beats they feel, and then at the stop tone they're supposed to stop counting and they report the number. You do that over a whole number of different intervals, time intervals, and then you take the average of that, right? And it turns out that although this task is very handy and people really like doing it, so it has a sort of major advantage, it's fast, it's easy, et cetera, and participants don't find it particularly onerous. The major disadvantage is that it's very hard to interpret this, and it is fairly clear it is not just a measure of sensitivity, despite the fact that sometimes it has been talked about as being an objective measure. So let me say a little bit more about why they're not likely picking up the exact same construct. First of all, these tasks only correlated about 0.32, which is not something you would want if you assumed they're exactly the same construct. Beliefs about your heartbeat can influence how well you do on the tracking task, okay? And what that suggests is that the internal model's playing some role in this task, right? Or at least it can be playing some role in this task. And I think the most interesting data set is this really interesting study from Winman and colleagues showing that if you surreptitiously pace people who have pacemakers and you don't tell them that you're changing their heart rates, their tracking answers do not track those changes in heart rate. So what this suggests is that we have to be a little cautious about how we interpret the tracking task. And because it has this sort of impact of beliefs, we have to consider the possibility that it's, at least for some people and in some circumstances, it may be as much about the operation of the internal model as it is about whatever viscerosensory signaling is coming up from below. Now, as you all are well aware, and actually have fostered, frankly, interest in interoception has exploded in the literature. But because of these measurement and construct issues and the various difficulties of doing this kind of work in humans, it is sort of slowing things down. In addition, I think we have to be cautious about when we choose a measure and say that it means X in terms of the construct it's reflecting. And so I think that's something where we could, as a field, probably be doing a little bit better. So I've proposed that the interoceptive sense data inform the brain about what's happening in the periphery, and that this is likely a major source of affect. And a predictive processing view suggests that these interoceptive sense data are part of what reshapes a person's internal model over time. And because the sense data are vague or ambiguous in quality, the brain can make meaning of these interoceptive sense data in multiple ways. And they'll do that differently across contexts, and they may do so particularly strongly as their personal history varies from someone else's personal history, right? Because that personal history helped shape that internal model. And from this perspective, I think a momentary experience, or the meaning that we make in a given moment about the current viscerosensory array, will vary probabilistically across different events. And this implies a really large role for context in meaning making. And it means that we need a scientific approach that enables us to capture the full, rich potential variation in affective experience. And that suggests the need to try to conduct, where we can, naturalistic studies outside the lab. And I'm going to tell you a little bit about that kind of work today, but I always like to show this particularly nice example. This is some work from Frank Wilhelm and Paul Grossman that illustrate just how affectively tepid the lab can be. So this woman spent several hours in their lab, exposed to a variety of stressors, and then she goes home that afternoon. She's wearing an ambulatory monitoring device, and at home she watches her favorite football team. Okay, now on the slide it's going to show soccer because this data was collected in Europe, but what you can see is that during the game she shows this much larger and more sustained heart rate response than any cardiac response she ever showed in the lab. And anecdotally, I don't think we have to look very far to find variation in our affective or emotional experience. In our daily lives, we can think of a lot of such experiences. So consider the nature of the anger you feel towards someone who cuts you off in traffic. That might not happen here in Tulsa, but I can tell you it definitely happens in Boston. Compared to the anger you feel when reflecting on the current state of politics, or the anger you feel with yourself when you've said or done something wrong. In all of these cases, we use the same label, anger, but the experience is distinctly different. And indeed, decades of studies suggest that affective variation is the norm. In fact, we see this variation in meta-analyses of autonomically mediated changes in peripheral physiology, in studies of brain imaging and single-cell recordings, in systematic reviews of facial muscle movements and how those are used in expressing emotion and also in perceiving emotion in others, and in studies of the mental features of affective experience, like valence and arousal. So historically, many emotion theories have assumed that emotions represent discrete physical types, where consistency and specificity of expression, experience, and behavior is the norm. And here, variation is typically treated either as error or it's ignored altogether. However, working from a constructionist theoretical view, I would say that variation is very meaningful. And this variation is created when physical responses, that is, actual or anticipated changes in metabolic need, are then optimized for a particular function or action based on a person's past experiences in similar situations. And by this account, emotion categories and the words that label them refer to populations of variable experiences that depend on both the person and the situation. And this implies the need for a deeply idiographic approach to studying affective experience, which suggests that there may be very situation-specific peripheral physiological patterns for distinct instances of a category like anger. And in fact, those might vary a whole lot across people. Now, testing these theoretical ideas requires some methodological innovation. In your typical standard lab-based studies of emotion, what do? I do it too. We bring people into a sterile lab environment, typically all by themselves. We have them listen to music or watch a movie clip to induce emotion. And that artificially limits the variation. And then, of course, we can't assess that variation. So the work that I'm going to show you today is the first sample of individuals who are in an experience sampling study where we made a critical innovation. We used a context-aware, physiologically triggered experience sampling method in which participants were prompted to report on their affective experience, but only after there was a substantive change in their physiology. Here, we were using a cardiac rate change, and it could either increase or decrease. We were actually tracking heart period, so that's the timing between beats. And our goal was to really just selectively increase the likelihood that we would get an experience sample at a time when something affectively interesting was happening. And you can really think about this as kind of compared to what we typically do in experience sampling where we would just randomly sample, right? So this methodology, which we developed with a company by the name of Mindware Technologies, which is in the Columbus area. This is a company I've worked with for all 30-something years of my career. And they basically enabled us to kind of set this system up the way we wanted to. And it really gave us the chance to look at both situation and person-specific patterns of physiological responding over time, and then to try to map those to features of self-reported experience, like valence and arousal. And in this way, we kind of flipped things on its head by taking a data-driven approach to try to discover, rather than to stipulate, what the meaningful biologically based categories of experience were for each person. And by doing this across multiple individuals, we could then see whether these categories were similar across people. All right, so we had 3 primary goals here. First, to discover and describe individual variation in patterns of physiological activity in everyday life. Second, to examine how these patterns are associated with the mental features of affective experience. First off, using emotion word labels, which we had people generate themselves, and then also affect labels of valence and arousal. And third, to identify and describe consistency in patterns of physiological activity across individuals in our sample. And before I provide the results, I want to acknowledge the 2 then graduate students who co-led the first paper on this work. This is psychologist Katie Heyman and engineer Dr. Zulkarnain Khan, both of them now doctors. I'd like to also acknowledge that Katie had a huge hand in this next set of slides, which are quite lovely. As part of a larger study, we had 52 participants who completed this experience sampling protocol using concurrent ambulatory peripheral physiological monitoring. Each day, participants were outfitted with sensors and portable equipment to capture the electrocardiogram, but also the impedance cardiogram, as well as measures of their movement and posture using these little inertial measurement units. They wore one on the torso and one on the thigh so we could get posture. And they wore this all for an admirable 8 hours a day for approximately 14 days. Now, while they wore the monitoring device, they also received prompts on an associated smartphone app, which allowed them to respond what their current affective experience was. And the majority of prompts were triggered by sustained changes in heart period. Now, one important piece here is that if the heart period changed due to movement, we didn't allow that to generate a prompt. Because really, we were wanting to focus on affective changes that we thought were likely to enhance our experience sampling. And so it is the case that if affective change occurred in the context of movement, those would be lost here. We also generated 2 random prompts a day so that not everything was linked to a physiological change. And participants were asked to complete 6 to 8 experience sampling prompts each day, and at each point they provided their current affect just using this little slider. They told us about both valence and arousal. They also self-generated emotion words, and they gave us intensity ratings for those emotion words. And they provided a really brief description of what just happened. And this was so that at the end of the day, when we asked them to tell us a little bit more about what had happened in that moment, they had a little tag and they could remember, oh yeah, that was when the person, didn't fail to yield at the stop sign and almost hit me. Okay? And these are the kind of events we do actually pick up sometimes. In the moment, they also tell us a little bit about the current social context, and they did a 1-item decision-making task. Now, using the ambulatory physiological data, we used 6 cardiovascular features. We used, obviously, the interbeat interval, respiratory sinus arrhythmia, which is a measure that we take from the high-frequency heart period variability, also from the interbeat interval data. We also have pre-ejection period, so just the time it takes between when the heart gets an electrical signal to beat and when it actually begins to eject blood. Left ventricular ejection time, so how long does that ejection take? Stroke volume, the amount it spits out per beat, and the cardiac output in liters per minute. So how much are you circulating in terms of liters per minute of blood? And change scores for these features were extracted for each experience sampling event by subtracting the mean value for the 30 seconds after the heart period change from that triggered the prompt from the sort of baseline, which is the 30 seconds prior to the prompt. And to ensure the comparability of results across the sample, we excluded anybody who had fewer than 70 artifact-free events. And so that took our sample of 52 down to 46 people, and that's our final sample. Now, next we used an unsupervised machine learning approach to explore within-person patterns of physiological activity. And specifically, physiological features for each experience sampling event were submitted to a person-specific Dirichlet process Gaussian mixture model. And what's nice about this method is that it allows us to discover the number of clusters rather than to stipulate them ahead of time. Because one of our key questions and hypotheses is that people would likely differ in terms of the number of physiological patterns that they frequently had. So this method allows us to discover the number of clusters and make probabilistic cluster assignments to each experience sampling event. We also analyzed the self-generated emotion words that were used to label the events, as well as their affective ratings of valence and arousal. So when I show the data, I'm going to show valence along the x-axis and arousal along the y, all on one plot. Now, returning to the first research goal, which was to describe individual variation in patterns of physiological activity in daily life, we observed a wide range of patterns within and across participants. And as we predicted, participants varied in terms of the number of clusters that we discovered and in the specific patterns of physiological change that were represented by those clusters. Now, to illustrate these findings, which are a little bit hard to show, we basically selected an example participant whose data and results were average for the sample. Here's a plot of the results of the clustering analysis, so let me orient you here. So each physiological cluster is one of the horizontal columns. You'll see that this participant had 4 primary clusters. The 6 physiological features are along the vertical axis. The numbers in there are raw change scores, just because some of you might care about looking at like how big was a given change. But the other thing you can see is that the color scheme, so large negative changes, large negative effect size changes were coded as red. So the deeper the red, the larger that effect size, and the deeper the blue, the larger the positive effect size. Okay? And what you can see is that these 4 clusters are really quite different. There are also some events that don't fall into any of the clusters, and I've left those off for now. But what you can see is that there is a fair amount of variation here. Now, as a point of comparison, here's another participant. This person was also fairly average for our sample. And notice that this participant had 5 primary clusters of physiological change. And if you kind of eyeball across these, you'll not only see that this person's clusters don't really look anything like the person on the left, but it turns out that that person on the right looks almost like no one else in the sample. So this person had a very distinctive set of physiological patterns. And this really, I think, is a very clear illustration of the extent of variation in physiological change that happens during affective experience, at least as it's experienced in daily life. Now, a second aim here was to assess how these patterns were associated with emotion labels. And here we found that participants generally used the same emotion word across multiple biologically defined clusters. And they also used many different words to label events in each biologically based cluster. So I'm going to show you this in terms of a bubble plot. So this is the same participant I was showing you their clusters on the left. Okay, this is all of the emotion words that they used and how often that was associated with any of the physiological clusters. And what you see is this sort of common use of many different words to tag any given cluster, but also very commonly multiple clusters being associated with any given emotion word. And basically this shows a fairly low amount of either consistency or specificity of emotion word usage. Now, you might think, well, okay, maybe if then you instead look at the affective valence and arousal, you'll start to see somewhat more consistency. And the shorter answer is, nope, turns out that doesn't really carry the variance either. So on the right, you'll see scatter plots for each of the participants' 4 clusters. So this is their first cluster for this participant. So you can think of that as what's happening in this first column here, A. And basically, the dots where they're darker simply means that that event was more likely to be pegged probabilistically to this cluster. Okay, so the really dark green ones were strongly linked to cluster A. So here's cluster A, so just kind of get the map in your head. Here's cluster B, cluster C. You notice that there are fewer events, but you notice that there's no distinctive pattern here. And so taken together, these data suggest that patterns of physiological change also do not map very neatly onto either valence or arousal. Now, our third research goal, which is the one we are a little bit thin on being able to sort of say something strong about, but was to try to describe was there any consistency in patterns across individuals in the sample. And given how small our sample size is, this you have to consider a little bit preliminary. But we're currently working on collecting a second full sample, and as soon as we have that data, we're hoping to be able to assess the generalizability of these findings. But as a first step, what we decided we would do is just look across all the identified clusters to see if there were any broad patterns that occurred. And these are all of the top 44 patterns that happened at least twice. And it turns out they account for 66% of the variance in the data. So it does suggest there is this set of relatively common patterns. And it makes some sense because we expect that we're going to have some variation due to bodily context, right? And I will say that probably as a function of the sedentary culture we live in, many of our prompts occurred when people were sitting. So we think that posture is one of the key things that is structuring this data. But taken together, we see that variation really is the norm. And using unsupervised machine learning analyses, we can find that individuals vary both in the number of patterns that they have in everyday life, but in the nature of those physiological patterns as well. And consistent with prior studies and with reviews, we see limited evidence of any one-to-one correspondence between physiological activity and subjective experience. Rather, the physiological and mental features provide unique, non-redundant sources of information about affective experience as a whole, at least on an instance-by-instance basis. We do see some consistency across individuals in terms of the patterns of physiological change, so they're not completely idiographic and also not completely random. And really the goal of our next sample in our future work is to try to ask the question, what is it that structures this variation, right? Because presumably there is structure here, we just don't quite yet know what that is. I'm guessing that many of you can see the broad usefulness of this kind of a tool where you can use physiologically triggered experience sampling method because it lets you really deeply sample people using their physiology to tell you something about what's happening in that moment. And this method really lets you integrate measures of physiological, contextual, and mental features. And I think it's a really potent tool for investigating individual differences. And I think it can be used for a whole lot of different studying a whole lot of psychological phenomena in everyday life. Now, I also think that seeing this extent of variation in affective experience suggests that we have to take variation more seriously, at least as affective scientists. And it means we have to use research designs that let us see variation where it exists, and then to try to model the structure in that variation instead of ignoring it. So I'd like to next consider a little bit, very briefly, how the body and the brain are coordinated in the support of action. And so you can think about it from the predictive processing view that the brain's most important job is optimizing energy efficiency and to efficiently regulate the body. Now we need to think about how somatomotor action, skeletal action, viscerosensory inputs, and extraceptive sensory inputs are kind of all working together to coordinate what's happening. And a key piece of this coordination is moving the sensory surfaces to enhance their ability to sense what is expected to arrive, right? And we would call this active sensing. So I'm just going to show some very preliminary data on how bodies and brains temporally coordinate visceral systems with the actions of the visceral output systems that support them. So, as you're probably aware, this is also a burgeoning literature where people are starting to look at how we coordinate peripheral physiological signals with perception, with learning, with action, and even with the inhibition of action. And in fact, I just this last week saw this new review out from Karen Roloff's lab where they discuss the cardiac effects of perception, and they do so within a predictive processing framework. It's pretty interesting. So the work I'm going to present next is being done by graduate student Kieran McVeigh, and this is in collaboration with Dr. Alex Galvez-Pohl. And in our review of the literature, we found that relatively few studies have actually ever looked at coordination across multiple viscerosensory systems. And that's where we thought we could probably add something. And in fact, it just seems strange to us because we know that the respiratory system has a huge impact on the cardiovascular system, right, in terms of respiratory sinus arrhythmia. And so it stands to reason that respiration, heart rate, and action should all be temporally coordinated in supporting active visual sampling, at least when the person is able to self-pace the task that they're working on, right? So keep in mind that this means in a lot of psychological studies, we aren't going to see this coupling. And why not? Well, what do we typically do in our studies? We present stimuli randomly, and that makes the occurrence of the stimuli unpredictable, right? And so what we're filling our experiments with is a lot of prediction error for folks. They just can't know what's coming. Now, probably they're making larger mental models of the entire experiment, right? And this is why sometimes we see effects when we block stimuli. But real life isn't really filled with the kinds of random appearance of stimuli, right? Instead, daily life is filled with a lot more predictability thanks to our prior knowledge of similar situations and contexts. Now, in this study, unfortunately, I only have about half of the sample so far. We have about 24 people, and we're aiming for a sample of 50. But what we're going to do here is look at respiration, heart rate, and eye tracking so that we can record how people are both fixating and saccading as they're doing a visual sampling task. So we do all of these measures at a fairly high frequency. We have 150 trials. We give people a small incentive just to keep paying attention. And we get a mean performance of 85 to 95% correct. So this is not a hard task. And our current N is 24, and we're gonna double that. And here's the question they're faced with. How many differences are there between the two arrays? Okay, here's the task. You're supposed to go back and forth between the two. You can choose whatever strategy you want to do that. Just tell me how many differences there are across the two arrays. And after scanning them all, they basically just answer a true-false question. Were there 8 differences here? Is that true or false? False, right? There's 7 differences here. So basically our goal here was to examine for all the trials how these two oculomotor actions, saccades and fixations, were aligned in time with the respiratory cycle and the cardiac cycle. So consider these data illustrative, again, because it's half the sample. So we're going to show these using circular plots. And saccades across the respiratory cycle are shown on the left with fixations over there on the right. And the peak of inhalation, or the end of inhalation typically, is at the 0 degrees at the top, with the end of exhalation at the bottom at 180 degrees. And in this initial sample, we actually don't see anything significant when it comes to saccades, but I will say it is kind of intriguing that, I don't know if you can see that very well, but this one big wedge here is consistent with some data from Park et al. From 2020, where they also showed that people tended to saccade right before the onset of an inhalation. So it's interesting, but again, this is not where we're seeing anything significant. That finding over there that people tend to fixate just before the end of inhalation is where we're actually seeing at least a trend. This is now the cardiac cycle, so the same thing. Saccades on the left, fixations on the right. We grade out the approximate part of the cycle that is systole. That's a little hard to do because systole is going to vary slightly from cycle to cycle, but the R spike is aligned at 0 degrees, and that gray section is taking into account the fact that there is some time between when the R spike triggers the beat and when the beat actually begins, right, and the pulse begins to get into the periphery. So systole is the gray wedge, and diastole is the whole rest of the black section. And again, with the cardiac cycle, we don't really have anything interesting going on in terms of saccades, but we do see, again, a trend toward that fixation to occur right before the onset of the next beat. So the evidence of coordination of body and brain is quickly expanding. However, again, limitations in our methods for assessing interoception need to be addressed. The folks here are certainly doing much of that really important work. And if we take a predictive processing view where allostasis and interoception are crucial in supporting brain and body coordination, then I think we have to try to broaden our designs to get beyond the randomly presented in-lab stimulus response paradigms, which have their place, but probably need to be coupled with some paradigms that take us out into the world and give participants a little more ability to act on their environment. And I think this work demonstrates that we can learn something important from more real-life paradigms that can allow us to model more naturalistic features of human behavior. I'm going to quickly thank past and present members of my lab, which I direct alongside Dr. Lisa Feldman-Barrett. I'd also really like to thank the funders of this work. So the work that most of the work that I showed you with the experience sampling was funded by the Army Research Institute, who continue to fund the second sample as well. And I would be delighted to take any questions. Salvador.
Thank you for a great and very clear presentation. So I was wondering, as I was listening to your presentation, one would anticipate that there is a major factor introducing variation, both intra and inter-individual variation, which is circadian rhythmicity.
Yep.
So I was wondering whether you put that into consideration. I'm not even talking about time of the day, because this being probably like a college population, I think.
Yeah.
You know, current use of social networks has made a diverge time of the day from circadian rhythmicity of interoceptive and visceral variant.
Right.
So, especially talking about prediction, I mean, circadian rhythmicity is basically predictive homeostasis.
Right.
It anticipates things that are not happening yet. So
Exactly.
I was wondering.
That's a great question. So for those of you, if you couldn't hear, it's basically a question about sort of what is the role of circadian rhythms here and have we considered that? The answer is you're absolutely right. I think circadian rhythms are going to be a huge part of what structures the variation. Here, we don't have a huge ability to access that, but because the way the technology currently works, we actually have to instrument everyone. So they come to the lab in the morning, they get instrumented, they get sent out into their daily life. When either the battery dies or they get sick of it, or they've done their prompts, they finish, usually in the early evening. Then they have to do the end-of-day diary, which is the part they really sometimes don't like because it's already been a long day. And then they're doing this for 2 weeks. So what we really have is what's happening during waking hours, right? And in part, that's because our interests are in, trying to also have these subjective mental features as part of what we're doing, which are going to be difficult to get at certain times of the day. But I think your point is well taken, that we do sample, a good 2/3 of the day. And we could look at whether there's kind of any sort of trajectory or cyclicity in that. And we have not yet done that by the time of day. But it's an interesting question. What we have looked a little bit more at is what are some of the things they're actually reporting that include things like their social environment. Am I on the computer doing stuff? Am I trying to find information? Or am I at dinner with friends? And so we do have some of that. And unfortunately, in this 42-person sample, there's just only so much you can do with it, right? We're hoping by having this other 50-person sample, which again is, you know, given how hard it is to do, 100 people still isn't going to be a huge amount, but it'll let us, I think, start to try to ask that, you know, because food, like we hope that some of what we're getting is I'm just sitting down to eat, so that we actually do have some allostatically relevant behaviors that we can even start to look at. But again, it's going to take a fair amount of sample to start to unpack that. Yeah. No, you know, I don't think that the ambulatory device, it does get ambient temperature. And I'm trying to think where it's measured from. I think there is temperature, but we don't include it here. Yeah, that's an easy thing to add. Yeah. Yes, yes.
You could get temperature if you paired this, if there was a way to integrate it with, there's this smart pill.
Oh, yeah.
Medtronic, that it's an ingestible pill that gives you body temperature, but also gives you like esophageal pressure, gastric, and acidity. Obviously, temporally aligning things together. But
that'd be awesome.
There are additional ways
yeah.
That are out there that are FDA sort of approved for measuring multiple internal signals.
Yeah, and that's the kind of temperature we'd want is,
yeah,
yeah.
So Ryan and then Mark.
Okay, Ryan. Yep. Features of the context?
Yeah, so for instance, and I know I think you kind of halfway answered this in the previous question, but just got like messy in terms of having sufficient number of constant context to look at.
Yeah.
You just might expect given the high level semantic nature of these things, that context might be better predictor than others.
Yep. Yeah. So let me answer your second one first, because I already blanked out on the third one, or the second one, trying to hear. So your question about will people use the terms in a context-sensitive, specific way, kind of irrespective of the physiology? Yeah. So that wasn't our original question, but it certainly is an interesting question we could ask. And what that would at least tell us is something about the extent to which context, which we would think it would, drives the concept you choose in the moment, right? And interestingly enough, too, the other tricky variation to deal with here is we have one person in the sample who I believe only ever used 7 emotion terms over 2 weeks. And we have another person who never used the same word twice, something like 147 different feeling terms. Now, they sometimes used feeling in a very broad sense, so we got peckish and hungry and calm and tired and things that, you know, for those of us who are strictly emotion theorists would say, well, it's not quite an emotion, but it is a feeling, and we allowed that, right? So we could look, and I think you're exactly right, and that's exactly what we would also predict, is that context is part of what's driving what cue you use, and so whatever emotion's coming to your mind as your current experience. Absolutely. So I would assume that, in fact, is something that we could do. So go back to your original question, too, unless I didn't fully answer that. We do. So, yeah, the trait-like emotional granularity stuff is a little tricky because the usual way it's measured, right, is you give people a list of adjectives, and you give them the same list, and you'd like to do that at each experience sampling moment, but we didn't want to do that because we wanted them to self-generate the experience. So what we did instead is in the end-of-day diary, they do actually, for a certain number of events, give us those adjective lists. so that at least we can get a measure of emotional granularity from that. But interestingly enough, we also do have some suggestion that based on that kind of data, people's emotional granularity improved from the beginning of the 2 weeks to the end of the 2 weeks. So even this measurement, right, is not innocuous. And what we don't know is it anything about the experience sampling? Is it about the end-of-day diary where you're doing quite a bit of writing about what it is that you were experiencing for a few of the events during the day? It's probably some combination of those. We just don't know yet what's the active ingredient, but that in and of itself was sort of interesting. So there is still stuff to be mined from this. And at this point, it's very much a proof of concept, but I think there are lots of interesting questions.
Yeah, you can almost think about that as a potential intervention.
Yes, exactly.
My first question was very brief, was just whether or not, if you got any measure of how clear the different clusters got.
Yes, there are some measures that we tried to do, but basically one of the things you can sort of do is you can ask the question, how likely is it that a given event is probabilistically assigned to one cluster versus another? And there were quite a lot of cases where the probabilistic assignment was very high to one cluster and not so high to other clusters. So it clustered a little more distinctly than I think I would have thought. But of course, this method lets us do that probabilistic assignment, so we could dig into that further. And in fact, I think some of the details of that are probably in the supplemental materials for the paper, so I can send it to you and see if you think that we've assessed that as well as we can, because there may be a way to do that even more. Yeah.
Yeah, so I was curious, looking at the phasic relationship between breathing, heartbeat, and saccade.
Yeah.
I'm trying to understand, what was the initial thought about that in terms of when and when not during those cycles you would expect?
Well, I don't think the literature is very clear on that yet. So what I wanted to see is, could we see what would happen if we do kind of a three-dimensional cube where we look to see for, are saccades more likely to occur at some point with respect to respiration, and then at some more likely spot with respect to the heartbeat, right? And is there anything about the kind of the heartbeat and respiration, which themselves are tending to be locked in a certain way, that makes one of those kind of the stronger driver, let's say, of one of these oculomotor behaviors. So the problem is this literature right now is kind of skimpy. Like there's some stuff on respiration and tactile sensation. There's some stuff, but these are all papers within like the last 5 years. And there hasn't yet been a kind of a nice overarching theme about what would exactly we'd expect. So we're sort of at the point still, from my read of the literature, of kind of seeing what the empirical literature says and then seeing, okay, now how does that shape what we think might be the theoretical frame for that? But it simply made sense from my perspective that these would all be tied, but at this point, we don't know exactly how.
So I'm going to just finish with one brief question for you, and then
Yeah.
We'll close. So thank you so much for a fabulous talk. I think that the sort of assessment of emotion in the wild, right? You've sort of clearly illustrated how to take the organism and assess sort of interoceptive variables in the wild. But what's missing, I think you'd probably agree, is what's happening in the brain, what's happening in the nervous system, and do you think we'll see a time where we might be able to integrate
Yes.
mobile EEG or
Yes.
FMERS or other neural measures that might enhance your ability to identify specific patterns? Do you think that's possible? And the second thing is, have you thought about taking advantage of this real life setup to then also induce affective manipulations? For example, like texting somebody, you just won $200, right? Like you could induce a positive,
Yep.
positively balanced response. Negative valence might be a little bit more tricky with IRBs and stuff, to see sort of how the organism's physiology responds in the wild.
Yes, because then what you're basically doing is you're bringing the lab to the world. Right. So yes, we've definitely thought about the possibility of doing that. I think part of the trick is, as you said, IRBs don't like it when we do things that are potent to people. But the thing is, very potent things happen to these folks in their everyday life, right? So what we're doing is considerably less potent. But yes, I think ultimately, eventually, we will get a chance to do those kinds of interventions. And that should be imminently doable. To your other question, we are absolutely interested in doing that, particularly with mobile EEG. So one of our colleagues, we have a decade-long group that meets called PEN for psychologists, engineers, and neuroscientists. Very clever. But basically what we did is we have for the last 10 years been meeting together. And it has been through that group that we've been able to do this kind of work, because I'm not somebody who can do machine learning and et cetera. But we worked together, and in fact, Katie was trained by Zulkarnain on how to do this sort of work, and she then learned about network analyses and other things. So we're sort of helping psychologists become more modeling savvy, and modelers become more savvy about the psychology and the theory. And that has led us to have collaborations with people like Dennis Erdogmas, who does do mobile EEG. And so at some point, we would like to link those two together. The piece that still gives me pause, and you probably saw it from that picture before, but it's much like me wearing this, only our device is even bigger than this silly thing, right? And so you're walking around in the world, and right now, I will tell you, that device is wired. Okay, so I told the first time I was pilot testing it, and I went to walk into my child's school, who at the time was in elementary school, and I thought, they're never going to let me in this building. They're going to think I'm coming to blow up the place, right? So we really have to get our tech to something that is less obtrusive, less obnoxious, and that's going to be a trick when we start to add those measures of the brain in, right? Because then you have all the social stuff that comes with looking odd in the world, and how that's going to impact your behavior. So yes, we're absolutely thinking about that, and we'll probably have to think carefully about how to minimize the social impact of that.